fifth bioelectrodynamics webinar today with Andrea Markels. So before we start, let me give you a brief announcement. So this webinar is supported by Engineering in Medicine and Biology chapter of Czechoslovakia section of IEEE, and also by International Union of Radio Science, URSI. I would like to give you some brief instructions. So I would like to ask you to switch off your video and keep muted during the talk. If you have any questions, you can either type them to the chat, if you can find it, or you can raise your hand after the lecture, and then we will go to the Q&A session after the talk is over. I would like to announce that the talk will be recorded, including the Q&A session, and you can find them later on our YouTube channel. I will send an email to all participants about the link to the recording. The talk will be roughly 45 minutes. And now I'd like to introduce our speaker, Andrea Markels. She's currently a Motilal Osrodky Professor of Physics at the University of Buffalo, State University of New York. She got her bachelor's degree in physics and applied math from the University of California at Berkeley, and her master's from Columbia University. It was in applied physics. Then she moved on to University of California, Santa Barbara, where she obtained a PhD in physics, which was on characterizing optical properties of semiconductors, heterostructures at terahertz frequencies. She was awarded then National Research Council Fellowship to initiate the terahertz program at National Institute of Standards and Technology in Maryland. Afterwards, she was a postdoctoral fellow at Bell Labs. And then she began her appointment at the University of Buffalo. It was in 1999. And since then, she worked up to the professorship, and she's one of the, I would say, top researchers in the world in the terahertz spectroscopies in biomolecules, I'm very glad we have her here. And to mention furthermore, currently, she has a very interesting funding from NIH, Department of Energy, and National Science Foundation in US. And also she holds an important recognitions as a career award from National Science Foundation, and also a SUNY Exceptional Scholar and Teaching Innovation Award. And she, importantly, did a great job organizing international conference on infrared, millimeter, and terahertz wave last year, which was great. It was really great to attend. Okay, so with that, I would like to ask Andrea to start her webinar, and I'm looking forward very much to that. So please.
Okay, so I'm just gonna Michael, so thank you so much for such a nice, flattering introduction, and I've known you for a few years. And we usually get to see each other at least once a year at a conference. And hopefully we'll be, all of us will get to see each other in person soon. And so I really thank you for arranging this so that we can still get together at least once a year. So I didn't download the app for Microsoft Teams. So I apologize for that. So unfortunately, so once I do start sharing my slides, I will not be on the screen anymore. And so there will be times when I might need to, use hand gestures, and I will unshare, look at you for a little while, wave my hands at you, and then go back to the actual presentation. So I hope that won't be too crazy. All right, so let me go ahead and share. And let's see if this will work. Okay, so thumbs up. You can see can you see my screen?
Yes, we do, perfect.
Super, okay. So, this talk is a slightly modified version of a talk that I've given before, but but I've been told in the past that, I don't always, I get too bogged down into what's interesting to me at the moment. And I don't always express the details of, why I'm doing what I'm doing. And so hopefully I'll address some of those issues and really encourage you to embrace the complexity of biomolecules, and try new techniques to address that complexity. So specifically, what we're trying to do is look at the biological impact of structurally defined dynamics. And, that title has actually been, evolving over many, many years. But I think that is indeed what we're doing. Okay. So I want to make sure that I don't forget to acknowledge the folks that really I'm going to be showing the main work of here. And so really what I'll be discussing is just focused on a very particular result, which has to do with catalysis, really, for proteins. And so I don't know if, let me make sure that you can see my,
yeah, we do see your full screen, if that's what you are trying to get.
Yeah, and can you see my cursor?
We do, sweet. I mean, at least
okay.
I do, yeah.
Okay. So Yanting Deng, so I should back up just a little bit. So Catherine Neeson and Mengyang Xu, this is Mengyang, and this is Katie, Catherine Neeson. So they were really responsible for a very early edition of the main spectroscopy system that I'm going to discuss the results of. More recently, Yanting Deng has, along with Deepu George, have made a new generation of that microscope, terahertz microscope, and so I'll be showing some results from that. And then our recent results with the new microscope use samples that were generated and characterized by Jeffrey McKinney. And I'm going to be discussing some computational results, which I think are really, really important. And hopefully that will spur some discussions with Dr. Cifra's group. And those results were with Alan Grossfield and Tom Romo. And they're at University of Rochester, which is very close to Buffalo. I'm at the University of Buffalo. And just to give you a quick localization of Buffalo. So many of you probably know of Toronto in Canada. We are just across the border from Toronto. Unfortunately, we can't cross that border. We haven't been able to cross that border for a year. Okay, is that this protein, and now I am actually going to stop sharing for just a second so I can use my hands. All right. Okay, so what you would be seeing there, right? Okay, I'm going to make my little molecule here. Here's my little molecule. Here's my protein molecule. So I have my polypeptide backbone, right? And then actually hanging off of that polypeptide backbone, now it's all the amino acid side chains, right? And so thermally, I have my system and it's excited. Hopefully you can see it wiggling and jiggling there, right? So the whole molecule is wiggling and jiggling, right? And it just looks like some kind of Brownian motion, right? And that would be from a molecular modeling, a standard simulation using a semi-empirical force field, but you would, even with a full ab initio quantum calculation, you would still get this wiggling and jiggling, okay? And so the question is, now I will go back to sharing. So the question is, with all that wiggling and jiggling, at the same time, we see incredibly organized motion in these extremely large macromolecules. Okay, so one of my favorite examples, we saw those wiggling and jigglings, right? Those random wiggling and jigglings, right? But in fact, if you look at, say, catalysis for an enzyme, and this is a particular enzyme that you're looking at with this movie. This is dihydrofolate reductase. And it takes dihydrofolate to tetrahydrofolate. Okay, that's the chemical process. And what you see is the NADPH, or NADP plus, I should say, NADPH complex with the enzyme. And then the substrate comes in and binds. So the big green object is the NADPH. The substrate, dihydrofolate, is in the orange, yellow and orange. And you see a proton transfer from the NADPH to the dihydrofolate. Okay, what you're also seeing is a large change in the structure, right, of this protein. Look at that. Look at how coordinated that is, right? So you see this binding, this flip-flopping of this white loop with that binding, right? So that the loop opens, allowing the NADPH to bind, do the proton transfer, and then it flip-flops back, allowing for that both the cofactor and the product to be released. So look how coordinated that is. So how can we imagine that those two things can be true? That you have this wiggling and jiggling, right? And at the same time, you have this coordinated motion. And this coordination motion is pretty efficient, right? It's not random access. If you waited for random access for these conformational changes, you'd have to wait many, 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 many years. So it's not some random access, right? It's coordinated. So why? How is it coordinated? So if you ask this question to a bunch of physicists, right, what they'll do, what do they do immediately? They'll make toy models, right? The spherical cow. And so you approximate what this system is. And so a very successful approximation is the elastic network model. In an elastic network model, you model this extended protein, right, the polypeptide chain, as a series of masses. So for each amino acid, you would replace that typically just where the C alphas for each amino acid are, you would say there is an effective mass. And then you would connect that to the other masses in the chain through springs. So you have a mass spring model, and you would do your classical mechanics for this system. And so Prochowski did this many years ago, I think in the 1970s, for example, for DNA, and came up with this very simple lattice model for DNA, and found some resonant vibrational frequencies for these masses on springs. Okay? And so you could do the same thing for a protein. And so here's DHFR. And there are a number of online apps which you can use to do this with elastic network models. And you'll find vibrational displacements, vibrations. And so here you see a couple of vibrations for DHFR. And so maybe there's these correlated motions throughout the protein, which are exactly these vibrations. And these vibrations enable this rapid access to these different conformations. Okay, so that sounds like that's a very physicist kind of point of view, where we basically ignore all the details and we just say, well, if I can reduce this down to this mechanical model, this is what would happen. Okay, now I have to go back to the other presentation. Hopefully I can do that, because I don't think we'll see what happens with the other movies. Okay, so if we have that nice simple model, okay, we can imagine actually, do we actually see those vibrations in proteins? Okay, so we could just, like I say, you can model that in that nice simplified way, and you can see these vibrations using this nice simplified way, and then you could actually measure those vibrations. Okay, so that is the physicist view, right? And so if you had like a simple system, like for a fructose crystal is shown here, you would see these nice, you could even observe those vibrations with spectroscopy, right? You could measure the absorption or the transmission of your sample as a function of frequency or energy. And you would see certain strong absorption lines according to those energies of those vibrational excitations, right? So you see this nice strong vibrational excitation for fructose. This is actually anisotropic measurement. I'm not going to talk about that. The point is that, this is what you might expect as a physicist. Okay. Especially if you're a physicist that is very used to condensed matter. If you're used to things like maybe even, for physicists, a molecular crystal like fructose is actually a pretty complex system, right? We like things like maybe 3-5 semiconductors, or even better things like just silicon and germanium, right? Those are nice, simple lattice systems where we can measure narrow vibrational lines. Okay, so that's ignoring the fact that this is a big complex system. A protein is a big complex system, right? And its structure, unlike this nice lattice model, like the DNA lattice model, where all the atoms are at specific points and they stay at those specific points. Maybe they do a small, very displacement from equilibrium, right? From those points. But, you have a well-defined, stable equilibrium, right? You have your minimum energy in your potential, and you have displacements around that. So for a protein, however, and look, I even have access to my laser pointer here, I think. Nope. Okay, so here's my nice complex protein. Well, the structure of that protein is pretty well defined, right? I can say that there's a alpha helix here and an alpha helix there, and these turns. In this particular case, there's a little bit of a beta sheet here. So I have these general structural elements, but the precise positions of the atoms actually vary, right? So here's my potential energy plot for projected along one configurational axis. And remember, so what does that mean? That means, one set, one vector in this 3n dimensional space, where n is our total number of atoms and 3 is for our 3 dimensions, x, y, z. So I have one, so if I project along one dimension of that 3n dimensional space, my potential energy varies, as you can see here, right? Lots of local minima, lots of local minima. So lots of configurations, atomic coordinates corresponding to a local minimum in our potential energy. So we have this structural variation, right, that is in fact thermally accessible. Okay, so the heights of these barriers between the different minima are on the order of kT, right? They're all thermally accessible. So all of these little valleys are occupied. And so that's a big difference, right, from our modeling for, for our solid state systems, or that original Proharsky model, right, for DNA, right, which says, oh, I have this lattice model, this well-defined lattice model with a well-defined relative atomic positions, and from that I can calculate my vibrations. So that would not be appropriate for these complex systems. And so when we think about that, well, what do we get now for these vibrations? Okay, and so my colleagues at University of Rochester and I participated a little bit on that, looked at that question. Okay, what about the vibrations for this complex system? And so what we did there was we said, okay, let's go ahead. Let me go back to this picture. So let's go ahead and calculate our vibrations for each one of these valleys. Okay, and again, this is just a single projection axis. So actually, we're going to calculate this for 800, 1,000 of these valleys. Okay, calculate the vibrations for each one of those, and see if, what do you get then? Do you get any specific vibrational frequencies or specific vibrational displacements that are dominant? Okay, and that we could actually measure spectroscopically. Okay, and so this is the overall idea. We calculate those vibrations using first you have a trajectory. Okay, so we have we take the molecule, we hydrate it and neutralize it, and then get it up to our room temperature and equilibrate that. Okay, and then go ahead and let the dynamics run, and then take some random snapshots of those dynamics, of those wiggling and jigglings that you didn't get to see except by my hands and the wire, but those wiggling and jigglings, take some snapshots of those, and then determine the instantaneous vibrational spectrum. Okay, now this has been done in the past, this instantaneous vibrational modes, or normal modes, instantaneous normal modes, but it was really focused just on the vibrational density of states, and really looking at what I would say is non-equilibrium states, not really looking at the vibrations themselves to any large extent. Okay, so in our case, we take those snapshots, we minimize the structure, we calculate the normal mode frequencies, the eigenvectors, okay, and from the eigenvectors you can get the dipole derivatives for those vibrations. And from those dipole derivatives, we can determine, so first, primarily we're interested in those eigenvectors, those structural displacements, because those structural displacements are the ones that are steering the conformation of the structure to that new conformation. Okay, but we want to have the dipole derivatives. Why do we want that? Well, because that gives us our connection to the spectroscopy, to experimentally measuring those vibrations. Okay, so we do those calculations, and then from the dipole derivatives, from those calculations, we can then actually calculate an anisotropic absorption. An isotropic absorption spectrum. Okay, so let's go ahead and do that. And so these were the results by Todd Romo and Alan Grossfield. And so what you see on the right-hand side is a whole bunch of curves, right? And you can see those curves have some strong peaks every once in a while and little peaks. This is the calculated isotropic absorption for each of those individual valleys, okay, for the vibrations of each of those individual valleys. Okay, and so that's the x-axis now is frequency and wave numbers. Okay, so I want to apologize. I do go back and forth between wave numbers, which is a standard spectroscopic unit, and terahertz, which is really the technological unit, right? So the people who are really working on creating the instrumentation, right, to do a lot of these measurements, right? They live in terahertz land. And the people who want to use that instrumentation for doing things like understanding structural biology and protein dynamics, they use the unit wave numbers. Okay, so what's our yardstick, right? What are our benchmarks between those two units? So one wave number is 30 gigahertz. Okay. And there's another community that is also important here. And this is actually the people who do scattering measurements, scattering spectroscopy. And they live in millielectron volt land. Okay. And so 8 wave numbers is equal to 1 millielectron volt. Okay. So, so what do we see here? So we see that these spectra have bumps and wiggles, and some of them have very, bright, dominant bumps and wiggles. So I'm going to point out this light green curve, the blue curve, and the, we'll call it orange curve. Hopefully everyone can see those 3 different curves. Okay, so you can see in those, so in the green curve, for example, you can see there's this nice spiky spike right around, say, 80 wave numbers. Okay, so there's, so this again is our isotropic absorption as a function of frequency, and this is for our vibrations and proteins. And so we might, if we just did a single calculation with a single starting structure for a protein, so sort of along the lines of the Proharsky type of calculation for a lattice of DNA, for example, we would see this nice strong line, and we would say, hey, look at that. That is a spectroscopic feature. We're going to go measure that. But maybe we had started with a different starting structure. And so let's just jump over to this orange curve, and you see this nice spiky spike right around 150 wave numbers. Okay, and so the person who started with that particular calculation, that particular structure, maybe from a crystal structure, dare I say, and let's see, yeah, the blue curve is actually from the starting crystal structure. But focusing on that orange curve, if you had started with that particular starting structure and calculated the vibrations, you would get this spike. Okay, you would be only considering a single valley in that rugged energy landscape that I showed you before with all those different localized minima. So, So you would say, okay, I'm going to measure a strong peak at 150 wave numbers. Okay, but in fact, your measurement will probably be on a macroscopic sample, right, with many, many, many proteins in your light beam, for example, if you're using light to do these measurements, or in your neutron beam, if you're doing a scattering measurement. So you're going to have many, many, many proteins in there. Okay, and each one of those proteins is going to be sampling a different valley within that energy landscape. Okay, so what will you measure? You'll measure that ensemble average, the average of all those different spectra. And that's that red line right there. Okay, so what does that say? So that says, at least experimentally, and maybe even more generally with respect to the dynamics of the protein, there's nothing special going on here. Okay, that you don't get any nice bright peaks. Okay, so what about this whole story of, okay, we have these wigglings and the jigglings, apparently random wiggling and jigglings, but we see this very nice directed motion in catalysis where we have this very concerted motions towards the binding, the substrate binding and substrate release, for example. So, so does this mean that our idea, right, of the structural vibrations of the protein direct those motions? Is that the end of that idea? So let me just talk about one more thing that was a result from the calculations, and then hopefully I will still have some minutes to talk about the experiments. So we have this huge, in fact, these calculations ended up with over 1 million vibrations, right? We're sampling over this entire landscape and we're calculating all these vibrations for each one of those local minima. And so at the end of the day, we have, 3 million plus vibrations. And they all have their own displacement vectors. They all have their own eigenvectors. That's the actual motion of the molecule, right? So we thought, well, just for fun, let's see what happens if we project a particular functional motion on all those vibrational displacement vectors for those 3 million vibrations. What would happen? Is there any kind of dominant type of motion that persists throughout that vast vibrational pool? And the answer, amazingly, is yes. Yes. Even though you've got all these many, many vibrations coming from all these different variations in structure, right? When you do that projection, and this is what's shown here, this is the projection magnitude. You see all those little dots? You've got about 3 million dots there. That's really fun to render the first time. It takes a little time for your little computer to plot all that. And what's shown in blue is a histogram. And you almost don't need the histogram for these lower frequency peaks. You can see there's some dominant projections. There's some large amplitude projections on that database. Okay, so that's saying that even, all is not lost. You don't have no structure here. You do actually have dominant types of displacements in your vibrations, even when you sample over the entire landscape, or a good part of it. And so that is very interesting and very exciting. But our isotropic absorption calculations were kind of a bummer. And indeed, if you actually try to do isotropic absorption of proteins with terahertz light, or far infrared light, as it used to be called, you don't see a lot of structure. So this is just a sampling of different proteins and the kinds of measurements that we've seen in our lab for isotropic absorption for cytochrome C, that's the upper left, photoactive yellow protein, that's the middle, upper middle, and hen egg white lysozyme, right? And I'm not going to go into detail of those spectra, except to say they're pretty smooth, right? They're pretty smooth. They don't have a lot of dominant features, right, that we can latch onto and try to understand specifically, how that vibration, we'd like to say, oh, there's a vibrational feature. Let's understand how that vibration impacts the biology. And, if you use isotropic absorption, you get nothing. So, or very little. Okay, so what do we need to do? Because we just saw, right? We just saw here that that in fact some motions do dominate this ensemble, this massively ensembled calculation. We see that a particular type of vibration does dominate, and it dominates at specific energies. That's what's really amazing. This is 5 wave numbers, and this is 8 wave numbers, those 2 lower peaks. And then the next one up, I think, is about 14, 15 wave numbers. So, so there must be somehow that we can get at this. But what pulled those features out was we did a projection on particular displacement. So we had to have a spectroscopy motion that had some kind of sensitivity to the structural displacements. All right, so in my lab, we've done the simplest thing along those lines that possibly could be done. And that is to look at the direction of motion. Okay, so one thing we can, if we take a look at a structure, and here's a little protein structure. This purple guy, it's decorated with some water. I'm not talking about water at all in this talk. That's a whole nother talk into itself. I would love to talk about it in discussion. So I'm just going to go through this fairly quickly because I know that I'm actually behind time already because of those technical errors. So very quickly, one structural discriminator is the direction of the motion relative to the overall orientation of the molecule. So for example, here is and that blue arrow is showing the dipole derivative or the dipole transition for a particular vibration. And, for this, charge system, that is generally along the effective direction of motion, okay? So you can think of this as kind of like a net dipole for the protein, and the vibration along with displacements along that line, right, gives us a net dipole transition or dipole derivative in that direction. So if I have a spectroscopic technique, which is just simply using the polarization of light, right, if I can discriminate, right, that direction of motion using the polarization of light, that's one way that I can pull out that structural information for that vast vibrational spectrum. And so that's what we did. So so I won't go through these animations to any detail. I just want to say, if I have the polarization one way, I can couple to that vibration. If I put the polarization the other way, I no longer can couple to that vibration. And so if I take the difference of those 2 measurements, I can isolate a specific vibration because of the direction of its motion. Okay. And so, so to do that, we have to have aligned samples, aligned proteins. We do those with protein crystals. This part I'm going to really scream right through. Okay, so we use protein crystals for a line sample. Protein crystals are really, really cool. They are completely different than the kind of crystals that many solid-state physicists are used to. The interaction between the basis of our crystal is completely van der Waals. There's no bonding, no actual bonding between the molecules within this crystal, which is pretty wild, right? And if you think about it, that what's going on there is just strictly dipole and van der Waals interaction, which is just really, really cool, right? You have this nice large charged molecules, right? And through that charge interaction, they actually align. Okay, so I'm not going to talk about that in any detail, okay, except to say that puts some constrictions on our doing these measurements. We had to actually build specific systems to accommodate these crystals, okay? So that's our near field terahertz microscopy, or I should say microspectroscopy. There's a question. And
do you want to answer it now, or should I wait later? I mean, I can unmute Cristiano, or if it's okay for you, Andrea.
It's okay. Okay, so I don't know. Is it okay? Because my question for you is you know, should we just make this more of a seminar style? People just can ask their questions as you know, as we go.
Yeah, I mean, I'm perfectly open. Actually, since it's so interesting, I'm perfectly okay as an organizer to go over time if it's okay for you. So yeah, I can unmute Cristiano.
Okay, yeah. Well, once we go ahead and do that. Yeah.
Okay. So, Chris, you can unmute yourself now. If you can do that, I cannot do it myself, unfortunately, but I allowed you to unmute. So
Cristiano?
Yeah. Okay, see you're muted. Or not. Yeah, let's go ahead.
Okay, I'll keep going, and hopefully that will work out later.
Okay.
Okay. All right, so right, so I won't go into too much technical details on this near field system, but you know, unfortunately, it is a barrier to access this overall strategy. Is that you would need to build yourself one of these near terahertz near field microspectroscopy systems. Okay, but if I encourage you, you know, anybody who's interested in it, I think you could do it much more efficiently than we have done it. I think that there's a lot of potential to move along those lines, given, you know, current you know, fiber laser technology, along with really wonderful electro-optic generation of detection crystals. Anyway, so we do use these crystals, and what's wonderful is that they give us an absolute alignment of the molecules, so we can actually register the particular spectroscopic features to a direction of motion for the proteins. The problem is that these samples are extremely fragile, and they must remain in liquid or be hydrated for the measurements. But those are technical details, and again, I would be very happy to discuss that with folks. So we've done a bunch of different versions of this measurement. Or this particular instrument. We had an early version that was made by Gigi Akpas and then later improved on by Katie Neeson. And it had wonderful results, and I'm going to talk about those results in a little bit more detail in a second. Then Yanting Deng did a couple of, and Katie did a couple of other measurements where, so those first measurements were rotating the sample relative to the polarization. We also tried rotating the polarization relative to the sample. And that's this polarization varying anisotropic microscopy. And that was a lot of fun because you can get really detailed spectra for different molecules fairly quickly. Unfortunately, those spectra are very difficult to analyze because you've added the birefringence. This birefringence really strongly affects the results. So we wanted to avoid that and actually be able to talk to our computational colleagues a little bit better. So we now have another type of instrument where the sample is stationary. We rotate the terahertz polarization, and we also rotate the probe polarization for electro-optic detection. And through that, we're able to do this kind of more efficient anisotropic spectroscopy. And for the, I was very, when we first saw these measurements, I'm very skeptical. And so I never quite believed the measurements until we did this instrument. And here we see 4 different crystals, and each of these crystals has a different thickness. That's kind of important for these measurements, to actually make sure that any kind of features that you see are truly due to excitations of your sample, and not due to some other artifacts, optical artifacts, typically multi-reflection effects. So by looking at different samples with different thicknesses, you can help to eliminate that question mark. And so this is just a series of different hen egg white lysozyme crystals showing you this reproducible, reproducible features. Okay, so that's a little bit about the experiment. So I want to go back very quickly to what new information have we gotten so far? So some of the new information we've gotten so far, I don't feel very confident in discussing, yet. Okay, so I'm just going to go to this kind of older story that had to do with lysozyme. So here what we did was we looked at free and inhibitor-bound lysozyme. Okay, so lysozyme is an enzyme, and here is the free lysozyme on the left, and inhibitor-bound lysozyme on the right. And the inhibitor here is what they call an orthosteric inhibitor. That is that it's binding exactly where the substrate would bind for this enzyme. And so what do we see? Well, here's our spectra. And let me go talk you through this very quickly. The lysozyme crystals that we were using here were really what I would call high symmetry. That is that many different symmetry operations can be performed, and you can still, get back to your original system. So this is a tetragonal symmetry group. So so the net information that we're getting here for rotating the polarization. So this is effectively, the x-axis is effectively the polarization direction relative to the 001 axis of this crystal. Okay, and so really for this full 360 measurement, this was a very early measurement. One of the things that was very important to us is just to see, yes, the data is doing what we expect it to do. That is, it should at least have 180-degree periodicity, and it did. Okay, so if you have a lower symmetry crystal, you would get actually more structure than you see here, but we won't talk about that. So the main point is that we see these nice bright features, these anisotropic absorption features, which is much different than those smooth spectra that I was showing you before from isotropic absorption. And those features dramatically change when you have inhibitor bound. Huge change in our anisotropic vibrational spectra. So, so this was a wonderful, that particular measurement was a wonderful collaboration with Alessandro Pacchiaroni and Andrea Oracini at the University of Perugia. and I know that I destroyed the Italian name, so I apologize for that. So they're experts in neutron scattering measurements as well as other things. And so they did complementary neutron scattering measurements on the free and inhibitor-bound lysozyme. And so that's what you see up here. And so on the left-hand axis, that is the vibrational density of states extracted from the neutron scattering. And so what you see is that there's very little change in the vibrational density of states from free to inhibitor-bound lysozyme. Okay, on the right-hand side is the difference between these two multiplied by a factor of 10. Okay, and so, you know, there is some bumps and wiggles here in that difference, but, you know, it's not an obvious thing. Okay, and then on the right-hand side is the anisotropic absorption measurements, where the black curve is the free lysozyme, the red curve is the inhibitor-bound lysozyme, and the green curve is the difference between the two. All right, and we just put it on the log axis, similar to what the neutron scattering folks do. And so, of course, you can immediately see there's lots of very distinct bumps and wiggles there. And that difference spectra on the right-hand axis is exactly the same kind of scaling as the left-hand axis. It's, you know, you just look, yes, it's very obvious changes. So the vibrational density state shows a net, maybe broad increase in bound lysozyme, but we don't see. So what do we see? We see all these bumps and wiggles, right? Whereas the vibrational density of states doesn't really see those big changes that we see. So why is that? You know, aren't we measuring the same thing? And it's no, we're not, because they're looking at the energy spectrum of the vibrations. What are we looking at? We're looking at the direction of motion. So we're not, so what do we see? We don't see a big change in that energy spectrum. We see a change in the direction of motions of the vibrations. So that inhibitor binding has what? It still has the same energy spectrum, but the directions of motions have switched, right? They've rotated, and they've essentially rotated out of the plane of the terahertz light. So that's really cool. So we see that we can actually see the modulation of the vibrations by binding. And of course, in this particular case, that was with allosteric, no, I should say orthosteric binding. And what's really exciting is that anisotropic sensitivity to the inhibitor binding also shows up in that ensemble average calculations. So going back to Todd and Alan's calculations, the inhibitor bound is in the green, the free is in the yellow. Okay, looking at the difference, okay, they have a difference with a peak right around 8 wave numbers, and that's similar to the difference that we see. We see a peak in the difference around 8 wave numbers. Let's see. So that's this difference here. Okay, so I note that this difference is the opposite of the difference that's being taken for the calculation. So I'll stop there. So the point is that, you know, isotropic absorption measurements cannot be used to identify structural motions, and that's just simply because unless you're doing a single molecule time resolved measurement, which would be very, very difficult. Maybe you could do that with Raman. I don't know. But it's really just coming from the fact that you're looking at an ensemble average of molecules that have a slightly different configurations, right? And so your vibrational spectra is actually different for each of those molecules, and the average essentially averages away any of those net spectral features. So by using the anisotropic approach or any approach, I'm sure somebody can come up with a much cleverer approach than this, which isolates the vibrations based on the structural displacements, right? Then you can actually isolate specific vibrations. Okay. Now, this is a quick seminar talk. So, you've seen that we can see a contrast with, for example, with inhibitor binding, but you can also see a contrast with temperature, photoexcitation. We're really excited about some photosynthesis proteins where we're at last seeing photoexcitation effects. And that these vibrations, these types of displacements, actually, if you do a calculation with robust averaging over the energy landscape, you can actually see that indeed the anisotropic resonances do persist with that ensemble averaging. So our current challenge is to do lots more calculations and really start to identify specific features based on those kind of projection analysis, right? We just do one simple projection analysis, but we'd like to, move on to really considering what we would call a basis of functional motions, okay? And now we also have a technique, what we call our SATM technique, our stationary sample anisotropic terahertz microspectroscopy, which really allows us to look at a lot of different systems and possibly use, different crystal symmetry groups or other means to isolate specific vibrations. So with that, I will escape and say hello again.
Thanks. This is really great. So maybe before you appear again, which I love to see you again, but maybe I have Michael speaking again here. I have some question related to your slides. So maybe I don't need to see them, but I'll take a
I can pop back. We know now that we can pop back and forth.
Cool, so great. So maybe I should be, first I should be polite and give a space to participants to ask, but because I have plenty of questions, of course. But let's try to see if something pops up from the audience. If not, I will start. I don't see immediately. So, there is finally, Cristiano is now taking his hand again. So Chris, can you unmute yourself? Cristiano Gallup.
Yeah, I was guessing on that you just jump out slide 14. That was saying something on water, the problem of water in your structure, and how does it interfere on the thing. I would like to hear more on the water.
Okay. All right. So first off, let's go. All right, many people already know this, but I'm going to throw it up anyway, just so we have something to look at together. Okay, so here is so a couple of things. First of all, if you don't have water, you don't have biology. So there's no. It is very you cannot justify looking experimentally looking at a sample that doesn't have water, okay? Because it doesn't, unless you're talking about strictly material science and not talking about biological impact, right? There's no point in looking at a sample without water. Okay, so you have to have some minimum hydration, and that minimum hydration is usually about 0.3 grams per gram protein. That's typically what is considered that critical hydration for the dynamics that are at least akin to the dynamics of a crowded cellular environment. Okay, so even at 0.3 grams water per gram protein, the water is going to strongly dominate the absorption. Okay, and so so part of the story for that anisotropic motivation was this idea that, you know, even if you could come up with some way to remove the water entirely, so let's say, and there have been protein systems where folks have been able to use other kinds of solvents, still have general dynamics, right? But they're not, you know, they don't have water anymore. And you say, okay, well, why don't you just look at those? Okay, and so that's the point of really what I was discussing today, is that, you know, even if you completely remove the water, just because of the complexity of the system, your isotropic absorption is still going to, you know, be featureless. Okay, but then on top of that, you know, most proteins of biological, you know, interest, right, must have water, and that water is going to add on to this smooth absorption spectrum. Now, what appears to be the case, okay, because we've been doing this for a little while now, is, you know, we are able to see these anisotropic absorption spectra for the proteins, right? And for various proteins, those are unique spectra. Okay, so you know, our hand-wavy thing is saying, well, you know, because we're doing a difference measurement, certainly the bulk water is removed from the absorption spectrum. Now, there are certainly water, there is certainly some water that is sufficiently associated with the protein. You might want to call it bound. I mean, there's certainly bound water. Without doubt, there's a few bound water. There's also that initial hydration layer, which is, you know, somewhat dynamic. There's exchange with water adjacent to it, but, you know, you have a defined water layer that is hydrogen bonded to the protein. Okay, that could be going along for the ride for these vibrations. Okay, so that could contribute to the total signal. With respect to the calculations, we included 10 angstrom water shell for the vibrational calculations. And I do believe that 10 angstrom water does contribute significantly. I mean, that's what we see from the calculations. And that is really in excess of the actual water layer for the crystals. Really, that would be about 7 to 8 angstroms between adjacent proteins. Okay, so within the crystal lattice, right? So between each protein within the protein crystal, there's no direct bonding. There's this layer of water. The crystals are typically about 50% water. And so the calculation actually includes really more water than is actually there for the protein crystal. But when we were doing the calculations, we did not do that as a crystal, because that's another order of magnitude, maybe a couple of orders of magnitude in complexity of the calculation. There's only, to my knowledge, only Dave Case is working on that if you know about somebody else who's doing protein crystal calculations, that would be awesome to know. But I really think that Case is the only one who's doing that, and I don't think, you know, his machinery is ready for general application. You know, he is applying his machinery to very specific questions. Yeah, so that's all I have to say about that. So with respect to the water, it's important. You must include it. From our perspective, you must include it. The bulk water, we believe we're removing just because we're doing this anisotropic difference measurement.
Okay,
So
thank you.
Sure.
Yeah. Could you, but with your kind of measurements, could you infer the quantity of bound water and the interfacial water? Could you try that?
I don't. I mean, okay, so the crystalline, I mean, it depends on what you mean by bound water, right? So again, if you're talking about the crystallographic water, of course, you know, that is resolved in the crystallography. In terms of that strongly correlated water, let's say, that first hydration shell. I mean, there are other measurements that try to get at that. Like Martina Havenit's work and Vinh Nguyen's work, where they do these concentration-dependent measurements and try to extract from the concentration-dependent measurements that effective hydration layer by the turnover, right? So in those cases, they're using an effective medium model for the protein molecules, the water shell. Okay, we'll call that the biological water. So I'm very hesitant to call it the biological water. Why am I hedging on that? And the reason is that I don't think that you can really think of it as a distinct layer. You know, I like to call it the hydrated protein system, right? So that, you know, it's like a semiconductor versus a doped semiconductor, right? So it's intrinsic. The protein has changed its physical properties because of that hydration water, right? But nevertheless, okay, so you can come up with this overall model of the protein, a water layer that's strongly associated with the protein, affected by the protein. Its dielectric responses change because of that interaction with the protein, and then the bulk water. Okay, and so using that, and so as you go up in concentration, right? Then you eventually exclude all bulk water, right? And so through that, you know, and you know the concentration, so you know the effective distance between the molecules, and from that you can extract the effective thickness of that water that is affected by the protein. Okay, that its dielectric response is affected by the protein. So that is probably a better measure of that, you know, bound water.
Okay, thank you.
Sure.
Thank you.
Sure.
So I see.
Thank you for the question.
Thanks, Andrea. I see Ogan Girl has a right hand. So Ogan, can you unmute yourself? Now it should work.
Sorry for coming a little late. I heard there were some technical difficulties. So my question was also about water. So you largely answered that, but to the extent that we often spread a broad brush around water, bulk water, and so forth, but there are various different categories or structural features to the water, whether it's in the inner hydration level layer or on a wider distance from the protein. And no water in biological systems, including in crystals, is ever really bulk water. And so I wanted to know your thoughts on that and what your perspectives, Andrea.
Mm-hmm. Oh, okay.
So
thank you, Ogun. Thank you very much for the question. So yes, certainly, and I agree. So in the cell, there is no such thing as bulk water, and I think that Martina's earlier results basically shut the door on that, right? If it, and I think that it's absolutely true that you have these. There's a lot of charge. A lot of charge in a cell, lots of charge, and so you've got first of all, you've got all the proteins, right? You've got all the nucleic acids, which are supercharged,
and you have a lot of hydrophobicity lurking in little corners as well.
Yeah, I mean, presumably that hydrophobicity is mainly encapsulated, right, by whatever the molecule is, right? That it's only showing its hydrophilic skin, right? It's not that it's protected. The hydrophobic part is protected from the solvent. But
that's why I mentioned lurking.
There's so much salt and everything else that's in there. To expect water to behave as you have in a purified distilled water. That's not correct. That would not be correct. And I think that is demonstrated by a lot of people are doing terahertz, trying to do imaging, tissue imaging with terahertz, and I think for the dominant contrast there is the different kinds of hydration that they have.
Right.
Sorry, go ahead.
Yeah, there are two implications. One is using this anisotropic or differential quality to the water structure, as you noted, as a means of imaging. The second would be to actually see if there were discernible water windows. So as you know, the microwave spectrum has water windows, and these in fact are used in a technical capacity, not just scientifically. Has anyone really explored in the terahertz range, in the case of biological water, where there are actual terahertz windows, where your beams and so forth can propagate more efficiently and more deeply, instead of being absorbed by the water more generally?
Yeah, I think there is some ongoing work along those lines. I think Alex Shirkanov has done a lot of work. Yuri Feldman also has done a lot of work more in the lower frequency microwave range. Those are two names that immediately come to mind. But I think that I think that the tissue imaging people, so there's Zachary Taylor, Emma McPherson, and Vince Wallace. That community, right, I think has definitely looked at windows for effective
transmission.
Right. Well, okay, so reflection. So the imaging, the imaging in transmission is just not going to be possible unless you're looking at histology slides, right? So in those cases, also Enrique Castro Camus also is working on this. So then you just can't get light very far into human body. So even though the water is different, that doesn't mean it's non-absorptive. Okay, so it's still very absorptive, right? So your penetration depth is still on the order of 50 microns. Okay, so typically the people who are working on this kind of imaging and looking for contrasts associated with hydration and different types of hydration, they're doing reflection geometry. So Emma, for example, has a TerraView system where you put the hand or whatever on a plate, right, and then they're reflecting off of that. Similarly, Enrique does this similar idea with looking. He has a wonderful application of doing diagnostics on the skin of feet for diabetic patients. To discern if there's any strong deterioration, and monitoring that in terms of.
But these are all TDS systems, right? My point being is that
for Emma for sure. Yeah. That's I am very hopeful that in the next 5 years even, that CW systems, compact tunable CW systems will be available, so that the kind of
that's
overhead.
The only way to answer that question, because if you're looking for specific frequency windows, it's difficult. I mean, there may be ingenious ways to do it, but it'll be difficult in a wideband probe to really understand it. You need to basically ratchet along different frequencies to probe each of those and their transmission or reflective properties.
Mm-hmm. Yeah, there's definitely a distinct opportunity for monochromatic sources.
Yes.
Definitely.
So
we are definitely seeing that. I can't discuss it too much, the details at this point, because they're relatively raw. But we're definitely coming to turn. We've definitely seen situations where the broadband excitation is actually complicating the result and the interpretation. Yeah. So hopefully in the next few years we'll see some. But the key is that they need to be tunable. They need to be tunable sources. So we're still seeing that. So the QCLs are getting really, really close. So we're very excited about that.
So what do you think, and I don't want to take too much time, but it's a related question. What do you think is the main impediment to getting these tunable, continuous wave, sufficiently powerful sources? I'm sure you're asked that question many times, but what's the latest on that? What is keeping us from making it happen, particularly in the sub-terahertz range?
Well, I'm not, I don't build devices. So I haven't done that for over 20 years, dare I say. Yeah. So I know, because how old my eldest son is. So the folks in those fields would be able to say that better. I mean, I know that in the past the issue has always been, relaxation mechanisms in the devices, right? are always going to have energies that that preclude, any kind of population inversion, right? So because these are at thermal energies, right? So that I think was an issue, but it may be in the past. So I think I've seen some, I don't know. I don't know.
Yeah, I mean, just for me too,
I know
it's
for
actually
us,
a mystery.
we're pretty excited about mixing sources at this point, so that we don't even have to consider a population inversion. And then it's a really materials game where you're trying to get around phase matching.
Yeah.
Okay, so that's. But I again, I'm probably I'm for sure not the best resource to know what is the key impediments to broadband tunability.
Another question.
There's another.
Plans?
Somebody else below.
Somebody else has a question?
Yeah, Louise. Louise, is that Nico?
Yeah, I see. Luis is unmuted. I'm not sure if he's having a question now, but Luis, if you are going to ask something, it's good time.
Sorry, I was not hearing correctly, and I was a little bit distracted just the last couple of minutes.
Okay.
See if you can repeat the question.
Yeah, do you have any question, Luis? Because you got
unmuted.
No. Okay.
Oh, okay. Sorry.
So I wanted to actually just quickly follow on. Any plans to do anything with COVID-related proteins, the spike protein, the ACE2 protein? Any plans around that? It's top of mind, obviously, for many people.
I would love to do that. I would love to do that. But what I would love even more is that other groups would adopt this overall technique, right? Contact us. We will help you to adopt the technique. We can even, we can consider other ways to access this type of spectroscopy with others. We very much would like to collaborate with others. And the reason that I say that is because, so many years ago, for my PhD, I did nonlinear response from 3-5 heterostructures. And, and I'm actually watching the nonlinear terahertz field, from the sidelines, and I'm seeing the same kinds of things happening there today as, what was happening, back when I was a PhD student with my colleagues. And what we would see, right, is we'd be after a particular effect. But, once you get into, nonlinear effects, a lot of different things can contribute. And to isolate and say, well, what is that actual mechanism? What's actually going on is very difficult, right? And that's for nonlinear spectroscopy. Well, here we have another side of that coin, and that is complexity. So we have a very complex system. We have actually very complex spectra. And so if we threw in a COVID spike protein, which would be heck of interesting, I'm not really sure, we could take a spectra. Okay, and I suppose we could publish that spectra, but I'm not sure what it would mean. What is its impact? How does that, get the ball down the field in terms of coronaviruses? I think that, it seems boring and not so sexy, but I think it's super sexy, frankly, is to, actually figure out how to extract the most information from the spectra that we already have. Okay, and once we come up with that protocol of understanding our spectra, or even dare I say, move on to even a more effective measurement technique, right? Once we get to that place, then we can go off and look at something that really has minimal information. For sure, there's lots of information on the COVID spike protein relative to, probably a lot of other proteins, for sure, right? There's been intense interest. But I would say that there's just been this flurry of publications that have really focused on, and for good reasons, how to eliminate its function very quickly, right? And disturb its function. So, that's my answer to that. Is we'd love to do that, but only if it is actually contributes. It can contribute in a meaningful way. And at this time, I don't see, at this very moment, maybe in a year, we'll be in a different place. But at this very moment, I don't see us, I think we would be more effective at focusing on more, better organized and well-controlled, and dare I say, less dangerous systems than COVID-19.
Okay.
Well,
yeah.
One idea that I was thinking is if there are some specific vibrations for the COVID spike protein, and to the extent that motions are relevant to function, and there are documented motions in the fusion and so forth of the membrane-bound proteins.
Mm-hmm.
But if there was some of these motions that were required for the literally mechanical entry of the virus,
mm-hmm.
that involve these vibrations,
mm-hmm.
then introducing either the
absolutely.
inhibitor
absolutely.
or introducing a electromagnetic countervailing field, almost like electronic countermeasure.
So I don't agree with that. So the first part I do agree. So I certainly think that the ideal would be that we would be able to, and this is true for the other proteins that we are measuring, that we are
not just COVID. General principle.
So I think that this type of spectroscopy, can be very useful for determining so-called dynamical hotspots, as I say, and where we could say, okay, you can have allosteric binding. You could have an inhibitor or promoter bound in this particular hotspot region that can promote or inhibit the vibrations necessary for that protein to function, right? For sure, right? We know that once the spike protein interacts with the cell, that causes the cascade motions to actually, break apart or break open the capsid and, deliver the cargo. Okay, so if we can inhibit whatever those motions are that convey that cascade of events, that'd be great. So that's okay. That I think is a useful way to apply the technique. But I would suggest that we need to get some of those fundamentals down to actually say, okay, this indicates that this is the dynamical hotspot that you should go after. Okay,
right.
And we need to get instrumentation to have real rapid throughput to determine that. Okay.
Mm.
So in terms of any kind of therapy based on terahertz, I am a skeptic. Okay. So again, because you've got this, how are you going to get that therapy to that specific place? Okay. I just, I don't see it. Okay. You're not going to illuminate the human body with, high power terahertz. That's not a good idea. Okay. So,
well, that actually raises a very interesting point about the distinction in medicine between medical disease and surgical disease. To the extent that medical disease, you take a pill, you take an intravenous drug, and it goes all around the body. And so I agree with you that terahertz is not going to be in this kind of category of medical disease. And I'm not speaking specifically about pills, but and medicines, but more about the distribution, the generalized nature of the disease. In contrast, you have surgical disease, which implies surgery and procedures, but more profoundly implies localized disease. So I would envision these sorts of electromagnetic interactions that might be trying to control protein functions and motions in a localized setting. So when you get a COVID infection, it's in your nose, it's in your buccal cavity, in your mouth, oral cavity. So there are some localized areas where if that process of infection can be reduced, that would have an effect.
Okay, so I think that the fact of the matter is that water is a great absorber for terahertz, and we we see this again and again and again, right? And that there's this great overlap in the vibrations of proteins, and that's any protein, with the water absorption. Okay, so I think that number one, you've got lots and lots and lots and lots and lots of different proteins everywhere in your body, and they all have that vibrational density of states that I showed you before, right? Mm-hmm. They all have that same vibrational density of states. The peak might move by 10, plus or minus, even I'll say even 20 wave numbers, protein to protein, but pretty much they all have the same vibrational density of states. And so if you cannot select out, it's a sad news, this is, doesn't help my proposals at all, right? It's a sad news. You cannot selectively excite this protein with that terahertz light and not excite that protein with that terahertz light. They're both going to absorb, okay? But what you can do is get information, right, about the functional motion. So again, this is using the spectroscopy to tell us about what are the motions, what are the dominant motions for that protein. But in terms of using that same light to, within the complex system, within many heterogeneous systems, shall I say, to use that light to excite and modify the dynamics of the protein, I think it could be done in isolation, but I don't think it can be done in actual living tissue. I don't see that happening. There's just, it's such a multi-component system that, you're not going to get that selectivity of this versus that. And that's what's the mission, right, of drugs, right, is to get both the high interaction but the specificity, right? Mm-hmm. And you can do that. I'm somewhat sold, maybe I'm around a lot of biochemists or something, but I'm somewhat sold that you can do that much more effectively chemically, right? Then you can do that with
energy. So philosophically then, at a meta level, drug therapy is also, you know, you ingest it, goes around the whole body. There's 30,000 different proteins, and in some cases you're looking for quite rare proteins, hormones or whatever, and miraculously it happens. Of course, there's side effects and things like that, but it is remarkable how drugs get to their targets and do their effects. But the same challenges seem to be the case as with electromagnetic modulation.
Well, no, I disagree.
What is the
very chemically distinct binding sites, right? So you know, the drugs are designed, you know, to dock at a very specific, you know, presentation of a binding site. And as you say, yes, there are side effects. There absolutely are.
And some of those side effects
bind to the wrong thing, but you know,
because there is some how should I say, a looseness in the binding. I mean, we have the protein dynamics itself. It's not purely lock and key. So, and some sites are very similar. So that there can be a loose sort of binding. So that's not entirely a perfectly lock and key system. I mean, everyone knows that that was a metaphor, but not exactly the true representation. So, in other words, there are similar challenges with the pharmaceutical paradigm, and there are similar challenges in the electrodynamic paradigm, but there seems to be some fundamental distinction between the two, which makes the drug side much easier than the electrodynamic side. That's what I'm repeating what you're saying. So I'm just trying to understand what is that fundamental difference, because they kind of share some aspects to this cross-reactivity.
I have a partial answer to that, if I may jump in.
Okay.
So exactly know again where you're heading to. Exactly, I mean, I would love to have these kind of tools. The thing is that currently the parameter space we can tune is more or less the frequency, maybe polarization. Some more things could be more fancy, like some quantum aspects. But the drug itself is like extremely let's say parametric space, because the location of every atom is already.
And you have nitrogen atoms, and you have carbon atoms, and oxygen.
So
the oxygen is negative, nitrogen is positive. So you have charge dimensions, spatial dimensions.
Exactly. It's very hard
stereoscopic
to
dimensions.
Fields.
So
macroscopically, we only have frequencies, intensity, maybe some modulation patterns.
Well, the beauty of electromagnetism is you have essentially one number, or a couple numbers. You have amplitude, frequency, and phase, and that describes everything, right? So that's simplicity.
Some more things you could add, but it's much more complex to deliver that.
Yeah, and so fundamentally a wave, you can have polarization and so forth, but generally you have a very limited number of parameters. Whereas in a drug system like you describe, it's a much richer landscape. So that a richer landscape, again, thinking philosophically, might offer more possibilities for minimum energy wells, that things can be stable.
I think Luis has something to say here, because
yes, please, because if you consider the chemical interaction between the drug and the receptor you cannot reduce it only to the number of carbons or number of nitrogens or number of hydrogens. It's the structure, the organization of the receptor that it's counting. And when you are speaking about the interaction of an electric magnetic field, I believe that it's the same thing. It's not just the interaction with the atoms, it's the interaction with the structure.
Yes.
Sense, you know. And so, I mean, the number of dimensions that you were speaking must be reduced, because if you have only atoms, you will not have no interaction of the drug, no interaction of the electromagnetic waves. I I am in the bioelectromagnetics since many years, and of course, there are people that says that this kind of just electromagnetic wave with that preferential frequency is able to change everything in your body. I don't believe this kind of things. Okay, but it nevertheless. When you refer to some structures, you can have perturbations due to the particular electromagnetic fields, yes, like the drugs, but not need a minimum of ensemble of atoms organized in a very precise structure to get that.
Yeah, I think that if you have a simplified enough system, for sure, we're hopeful that one could see some kind of resonant effects, right? Along the lines of so like I showed you in the calculations, right? 5 wave numbers and 8 wave numbers. And that was not at all apparent, right? That was not at all apparent in the spectra themselves. It was only apparent when you actually compared the specific displacements with the vibrational displacements, right? And in that case, it was the displacements associated with inhibitor binding. So those two frequencies, maybe you could actually in a simplified enough system, so you don't have too many different proteins running around, right? That you could actually affect the turnover rate.
This is exactly where our project is focused on, because knowing this lack of specificity in complex system, we decided to go to prove this concept in a simple, what we call nanotechnological system. We have few, let's say, microtubule or simple, let's say, one type of protein and another one which interact. And we want trying to see if we can use this for nanotechnological purposes, exactly to enhance rates of transitions and so on and so forth.
Right, exactly.
Where we could see something. And then
exactly.
There I have a couple of questions, but I would like to ask first, Andrea, aren't you too exhausted, or do we have still some time to discuss, because we had quite a long discussion already? Well, how are you feeling?
I did want, so I know that we talked about having a discussion with your group, just along the lines of the calculations, but I don't know if it's like so it's a Zoom era of lectures, right? So for my students, I say, well, at the end of lecture, I said, well, we're going to have a post-lecture discussion now, but you can go. It's okay. You don't have to stay. So whatever you'd like.
Trying to know the boundary conditions, physically speaking, for you.
Yeah, I would say, okay. So here, and also it's very late in Prague, but
it's fine. It's fine. It's also people from my group had to because we're already 1 hour overdue, had to leave. So it's anyway. I have some more general questions, because the technicalities we can do if maybe some.
We could do that. Yeah, that's true. That's true.
So I mean, maybe let's say on the level of, let's say, maybe less philosophical than Ogan had, but still more, I would say, still quite general things. So you already touched it. I was really following a lot the last the calculation paper which you published, and with this lysozyme anisotropic calculations, particularly those where you showed it on the slide, where you basically projected the spectra along biologically functional modes, right? So and there you see these beautiful resonances. I'm hunting also resonances since my start of the career in absorptions, because it's something non-trivial which could trigger particular function, right? So this is very philosophically I'm along the lines of what Ogan is trying to see, but to boil it down, we'll have to have a clear system to see that. So I'm curious, do you think that these rather sharp peaks could be accessible spectroscopically, because you can somehow see them, but what is your feeling about, like,
well, exactly. So we haven't set up that measurement, right? So we see some indication of the 8 wave number peak in the measurements that we've done so far. Now it's conceivable, okay, so those are the anisotropic measurements on the tetragonal crystals. Okay, now the tetragonal crystals, right, are this really high symmetry group. So we're actually canceling out a lot of dipoles. And so so it might not be the best system to look at. So we have been working on looking at lower symmetry crystals to see, you know, a more richer spectrum, and then possibly, you know, in that way, you know, again, do the inhibitor binding comparison to see if we could find them that way. Now, of course, what I'm more interested and excited about the possibility is using those specific frequencies to actually perturb catalysis. Yeah. So now the problem, okay, so that sounds great. The problem is that lysozyme is slow. It's a very slow enzyme. And so the practicality of that, you know, it might be kind of tough, right? Because, you know, if you're going to do catalysis, then you want to do it in solution, and then you're gonna blast it for, you know, the time that would be, you know, for the turnover rate for lysozyme, which is on the order of milliseconds, seconds. And so then you got a real heating issue. So so it's tantalizing, right? And so you're like, okay, so maybe this is not the most ideal system, but what do we see here? We see, you know, I don't think lysozyme is particularly special, right? So you know, so it's quite likely that, you know, those, so we talked about dynamical hotspots structurally. So we could also think of dynamical hotspots spectroscopically. So we could employ this same machinery to a more amenable enzyme, an enzyme where we wouldn't have to really think about, you know, the temperature concerns, the heating concerns, you know, would be minimized.
Okay, yeah, I see the point. That's actually very well. Of course, we'll be concerned for that. Another, like, let's say, big question, which is always thinking about, you know, it's typically people think like the lower you go with the frequency in the dielectric-based absorption, it seems to be that it's lower chances of having something resonant there, having water around, right? Typically, it seems to me like that. You know, so some of the slides you showed, it was not maybe in this talk, but when you switch between the PowerPoint presentations, you show that there are expected underdamped motions above 0.5 terahertz. So is there any particular reason for that in general, or is it something particular for lysozyme?
That was, okay, so that, all right, so frankly, there's just not enough calculations out there. So that was a specific calculation by Meinhold Merzel in Ljubljana, right? And Jeremy Smith, right? And that was a long time ago. And you know, Meinhold went on to different things, and Francie is doing different things. So so sadly, those have never been followed up on, and that was one of the few calculations that I could find that looked at damping, right? So I think there's not enough information out there, and the machinery is not open source. So we need, we've got El Nemo, and we have all these different open source, not open source, but open access computational tools right, for many other aspects of structural biology, but this is not. And right, so there's no surveys, there's no, it's one and done. And so, that's all I have to say about that. That was for Staphylococcus nuclease, I think. I doubt if let me see if I have that. I'll see if I have it.
It's fine. I'm just curious because also the choice of the line width, right?
Yes.
Also something related
absolutely.
to that.
Absolutely. Yes. So absolutely. So the line width, we arbitrarily use for those calculations. We arbitrarily choose a line width based on the results for molecular crystals.
Okay. So that was these.
Not because we calculated that line width, but that's it. So, Klaus Schulten many years ago also did a simple calculation with, the concept of the calculation is simple, and that is, just he put a large displacement, right? And I don't think he used it. It wasn't as sophisticated as taking a particular vibrational eigenvector and using that as a net displacement, right? And looking at the relaxation. It wasn't even that specific. But yeah, so things like that have not been done, to my knowledge, unless you know about it. I don't know about it.
We are trying to understand exactly these questions, right? Like, because this line width determines frequency selectivity and also the the Q factor of the resonator, so to say, and how much energy it accumulates. So it's absolutely essential. And therein comes the question is also we had, we're implementing now since last year polarizable force fields instead of because those should be better, represent better the electronic polarization effects, which can be very important. And of course, it's increasing the computational cost. So it's again a trade-off. So
yes.
And there could be, we don't know how that will affect potential, strengths of the resonances, but it's one of the things which definitely will be affecting terahertz spectra, just calculated by molecular dynamics using that. So, okay, going to these, intriguing resonance questions, the only papers I remember I've seen which show some probably dielectric-based resonances in nanostructures were on the viruses, actually. But they all come only from a single lab. So I'm a little bit careful, not to say skeptical.
Yes.
But this was like a good microwave engineering work behind. So I'm kind of trying, it looks like it's not unfeasible Q factors. Typically it's something like units and tenths, but at the frequencies of the few tens of gigahertz, which is very low.
Yeah, I mean, I can buy that, I buy that because, like you say, if, and I don't know exactly which ones you're discussing, but if you are in those low frequencies, and I can see that being commensurate with the capsid size, right? And you've got this dielectric shell. I'm a little surprised. I would expect it to be pretty well damped because, whatever the heck is inside there. But I could buy that that those isolated samples would have resonances. That's been, it's a plasmon type of resonance, right?
Might be. Actually, it was interesting that in those papers, I can maybe refer that it's to you, send it to. They've seen the pH dependence, which kind of makes sense because you're influencing how much charge you have. So probably they will some influence how much you bound the water. Maybe you make the outer layers of the water more elastic. That's kind of my conceptual speculation. If you have more charged interspace, maybe water is more strongly oriented, so it's more kind of elastic, I say. So that's something which is very interesting, and this, of course, is very heterogeneous, different proteins. So maybe it's maybe protein dependent. If you go back to the protein from the large capsid. So
right, but okay. So then, all right, that's really interesting. So Yeah, so if you change the protein, then you would, change the net hydrophilicity at the surface. You could think of it just as that, macroscopic effect. Also, it's really interesting to me, and I haven't seen again, this is probably my fault, is that the heterogeneity of the water over that surface, right? So I can totally buy that those resonances associated with the capsids. I totally buy that would be dependent on the protein composition of the capsid. I totally buy that, right? But not because of the dynamics within the protein, right? But just the effect that it has on the dielectric of the shell, as well as the interaction with the water. Yeah, absolutely. That's really interesting. That's really cool. Yeah.
Yeah, the authors interpreted it as based on the vibration modes of the whole capsid, which somehow, that was their interpretation. So, that was, if it was vibration based, I was a little bit, curious how it could be possible. But I'm a little bit just careful about those things, because it's
exactly.
Tried it. Another thing which it's interesting there in those papers, it's that since the viruses are it's homogeneous population more or less. So that's at least excludes some inhomogeneous broadening partially, because they are they are very similar to each other. So that's interesting. So, but it meant something to try. Okay, good. So we got reduced the attendance only to the, let's say, I would say hardcore attendees, or people who just have it playing as a background.